Dear students, now I am going to discuss about meiosis cell division. The term meiom means to reduce. Meiosis is a cell division that reduces the number of chromosomes in the parent cell by half and produces 4 gametes or gamete cells. So, this cell division occurs in germ cells during gametogenesis. Germ cells are the gamete producing cells, those are present in the gonads, that is in the testis and ovary. So, meiosis occurs in diploid reproductive cells. They are called primary spermatocytes of testis during spermatogenesis. Similarly, meiosis occurs in the primary oocytes of the females in the ovaries during oogenesis. During meiosis, diploid cells having 2n number of chromosomes undergoes 2 successive cell divisions. Meiosis 1, that is actually the reduction division, and followed with meiosis 2, that is the equational division, that is similar to mitosis, to produce ultimately 4 haploid daughter cells. They are having n number of chromosomes, which ultimately develop into gametes, sperms and eggs. So, in meiosis, DNA replicates or the chromosomal duplication occurs only once, followed with 2 successive cell divisions. That is what you can see in this cartoon. This is 46 number, that is 2n number cell. It undergoes meiosis first, it is the reduction division, resulting the n number of cells. These are having 23. Then further you can see the meiosis second, and that results into the 4 gametes, each having the 23 chromosomes, that means n number of chromosome. Meiosis is a specialized cell division where number of chromosome is reduced by half, creating genetically distinct haploid cells. So, meiosis is a reduction cell division, occurs during gametogenesis. This is spermatogenesis, what you see, and this is oogenesis, where diploid eukaryotic cell divides twice to generate 4 haploid cells. These haploid cells result into 4 gametes. So, this is what you will find. The meiosis occurs here during spermatogenesis. This is meiosis 1, primary spermatocyte that is having 2n number undergoes meiosis first to result into 2 secondary spermatocytes, they are haploid. It undergoes further meiosis second to give rise to the 4 haploid spermatids, which undergo differentiation to become 4 sperms. Similarly, you will be able to find this is primary oocyte which undergoes meiosis first. This is the reduction division, going to give rise to secondary oocyte. They are haploid in nature or haploid cells. So, chromosome number is reduced to half. Then this secondary oocyte undergoes meiosis second to produce the ootid. This is the haploid ootid you will find. So, first meiotic division is distinct from mitosis, hence it is called heterotypic division. Meiosis first is distinct, that is why it is called heterotypic division, whereas meiosis second is similar to mitosis, so that is why it is called homotypic division. But these division occurs continuously, so for our convenience we have divided meiosis into meiosis 1, meiosis 2, then meiosis 1 divided into the prophase first, metaphase first, anaphase first, telophase first. Meiosis second divided into prophase second, metaphase second, anaphase second, telophase second. Whereas here the prophase first is the longest stage in the meiosis. Sometimes in case of amphibia, it may span about 1 to 2 years. In case of human females, it may many years. So it is further the changes that occurs in the cells during prophase is significant, and it is divided into leptotin, zygote, packitine, diplotine and diakinesis. So in this you will able to find the stages of meiosis. It is you should see quickly. So this so this is leptotin, zygote, packitine, diplotine, diakinesis. This is metaphase first, anaphase first, telophase first, then prophase second, metaphase second, anaphase second, telophase second. It is quickly moving, and this is a picture that is real time meiosis, what you see in this micrograph. So this meiosis first, the prophase first is the longest stage, characterized by chromosomal condensation. As usual, you have seen this also in mitosis. Then another important one is synapsis, that is the homologous pairing. Then followed with crossing over, or there is a exchange of genetic information that occurs between homologous pairs. Then there is a appearance of a chiasma. All these are the events you will able to see in the prophase first. That's why prophase first is divided into leptotin, zygote, packitine, diplotine and diakinesis stage. So the chromosomal events in prophase first are coiling of the chromosomes, as I said earlier, that is because it is a typical of prophase stage. Then there is a homologous pairing. Homologous chromosomes are the homologous pair where the one chromosome from the contributed from the father and another from the mother. So one is paternal and another is maternal. So they are going to pair. They come lie side by side during the process called synapsis, and then followed with you will find there is a exchange of the chromatid segment between the homologous pair that occurs during crossing over process. Then after that you can see wherever there is a crossing over, there is a cross like structure you will able to see. That is what you can see, the chiasma. So these are the events you will see in the prophase first. We will study one by one in detail. In the prophase first, the first stage is leptotin. Leptotin meaning thin threads. During this stage, individual chromosomes begin to condense. So during condensation, what happens, you can see the telomeres of the chromatin, because they are very thin, that's why the telomeres of the chromatin attached to the nuclear membrane, you will be able to see, and they assume a stage like a bouquet stage, this is called. So this is the characterized in the case of the leptotin, it is the chromosomal condensation. The next stage is the zygote. That means the pairing threads. Zygo means pairing threads. There is a lateral pairing of homologous chromosomes. You will be able to see that they are showed in different colors here, the red one and the blue one. So they are homologous pairs. Now they lie side by side because of the development of a synaptonymal complex in between them. So now these paired homologous chromosomes are called bivalent. They have two chromosomes, appears as a single unit, that's why it's called bivalent. They may also be termed as tetrad because they consist of 4 chromatids. That's why the 4 chromatids going to form a complex, that's why it is called tetrad. The 2 homologous chromosomes become zipped or attached together due to the development of a complex. You can see it is synaptonymal complex. This process is called synapsis or pairing of homologous chromosomes. So the next stage is the packetine stage. The zygotine followed with packetine, meaning thick threads, further condensation of the chromosomes. So then there is the tight coiling occurs between the homologous pairs. Then it is followed with genetic exchange or crossing over takes place. So now you can see the blue and red, they are homologous pair and they are tightly coiled. You will find they appears as a single unit. You will find tetrad or bivalent, and there is a exchange of chromatid segment will take place between the non-sister chromatids, chromatids belonging to the different chromosomes. So non-sister chromatids undergo cut at the same level, they undergo cut at the same level and followed with the exchange of short segments of chromatids, and this is what you can see, the exchange of genetic information or DNA between two chromosomes. The exchange of information between non-sister chromatids results in a combination of recombination of genetic information, or else the new combination going to result. This occurs during packetine stage. So packetine followed by diplotine. After genetic exchange, paired homologous chromosomes, they repel each other. Once the genetic recombination is over, now the homologous pair, they separate. You will be able to see here there is a significant gap going to occur. They separate now after the genetic exchange, and they held only at one or more points all along the length of the chromosome. It appears like a X-like, you can see where exactly this is called the chiasmata, the cross-like structure, and the chiasmata are the regions formed by the breakage and reunion of non-sister chromatids. These are the region they are showing the where exactly the chromatid segment get exchanged between the homologous pair. Chiasmata number along with the homologous pair depends on the length of the chromosome. If the chromosome homologous pair is long, then you will find the chiasmata at many places, or else you will, if it is short, one chiasma per homologous pair. So the chromosomes are clearly visible under microscope during diplotine stage. Diplotine characterized by the presence of chiasma or chiasmata. Then this is followed with diakinesis. So the diakinesis, further shortening of the chromosome or condensation of the chromosome. Chromosome now looks very thick. So this is the last stage of prophase first. Chromosomes become highly condensed and dark and short structure, what you see here now. So this the nucleolus and nuclear membrane disintegrate. So this is the end of the prophase first. So nuclear membrane and nucleolus disintegrate. The homologous pair still held at one or two chiasmata. There will be there, chiasma will be there. You will find there will be two chiasma. You will find this is they appear in the form of X, or else in the form of, if there is a two, you will find two chiasmata, then you will find it is in the form of a O shapes. So all this typical to that of diakinesis. So after the diakinesis, the last stage of prophase first, then next is the metaphase first. So before that, just I sum up what happens during prophase first. In the prophase first, already duplicated the chromosomes, they pair, homologous pairing, you can see they lie close to each other. So that's why you can see the maternal duplicated maternal chromosome and paternal chromosome going to pair during the zygote. Then there will be exchange of chromatid, non-sister chromatid segments between the homologous pair, that you will find during packetine. Then after that they separate. So you will able to see in this image, you can see there will be a new combination occurs in the chromatid. So this chromatid later, each chromatid going to each different daughter cells. So the new combination appears when it is a new combination when compared to this is what you will find when compared to the parent combination. So this is what the changes that you see in case of prophase first. Prophase first followed with the metaphase first. So metaphase first, you will find the chromosomes in homologous pairs, they arrange on equatorial plate or metaphasic plate. So where you can see the centromere of homologous pair, they faces opposite poles. They are associated with the spindle fibers, what you see here. So the well organized spindle apparatus formed here, and the centrioles at this mitotic apparatus going to form. So this is the same thing, spindle apparatus you will form going to form even in the meiosis. So this is the spindle apparatus where you can see the homologous pair, now they face opposite poles. Next, it is followed with the anaphase first. The homologous pair now separate and move towards opposite poles. So this is the difference between the mitosis and the meiosis. Here the homologous pair separate, not the chromatids of a chromosome separates here in the case of mitosis, but here anaphase you can see the paired chromosome now separates. Each chromosome move towards the opposite poles. Segregation of chromosome or reduction in chromosome number now occurs, will result anaphasic movement of the chromosome. During anaphasic movement, the chromosome they form the V shape. So anaphase first followed with telophase first. The haploid set of chromosome at each pole now diffuse and organize into nucleus. Cytokinesis occurs and resulting two haploid daughter cells, we each having haploid number of chromosomes or n number of chromosomes. Two haploid daughter cells resulted in the meiosis first is now enter the meiosis second. So meiosis second is similar to mitosis. It is further divided into prophase first, metaphase first, anaphase, meiosis second divided into prophase second, metaphase second, anaphase second and telophase second. There is a small mistake you will find, this is prophase second. Two haploid daughter cells resulted from meiosis first now enter prophase second. Prophase second followed with the metaphase second, all these you have studied. There is a formation of spindle apparatus, then chromosome alignment in the equator, you will be able to see here in the metaphase second. The chromosome, the two chromatids you can see they face opposite poles. Then anaphase second you will be able to see the centromere cleave and the sister chromatids separated and move towards opposite poles, and the telophase you will be able to see the chromatid sets, the poles start uncoil and new nucleus going to appear, and cytokinesis going to result into 4 haploid the gamete cells at the end of the meiosis second. Thank you.